Good morning, HB 202 students. How is everybody doing? Now, before we get into today's lecture, I want to make an announcement. And the announcement concerns your student evaluations, your course evaluations. If you can fill in those course evaluations, and I'm assuming everybody's had the emails and everything else from the faculty, if you can fill in those course evaluations, and not only for this course, HB 202, but all your human biology courses, we would be greatly appreciated. And as an added incentive for filling in the evaluation for this course, what I normally do every year is the following. We have something called GPA boundaries. And if we hit the 75% student, in fact, I'll lower it down to 70%. If we hit the 70% student response rate, then the following will kick in. Number one, the GPA boundaries will be bumped up. In other words, the coveted 84s will go to 85s, 79s will go to 80s, etc., etc. And for this particular course, what I will do is take that 89% and bump it up to a 90. Normally I don't do that because I believe you have to earn that 90, but this is a special year, so I decided to make that available as well. In addition, I will instruct the TAs to essentially mark happy. So if they mark happy, usually the grades will go a little bit higher, etc., etc. Not only that, but we usually give them food and caffeine, so that should be okay as well. But that's only if we get to that 70% student evaluation mark, okay? Student response mark. If you have any questions about that, you're free to contact me from that. Now, on to today's lecture. This lecture is about patient 0. And specifically, it is about how this virus essentially was able to go and infect over 80 million people and kill 40 million of those people. Where did this virus come from? And many of you, if you Google patient 0, you'll come up with an individual who's Canadian. And unfortunately, that person in reality really wasn't patient 0. So we're going to investigate why this happened. We're going to find out exactly who patient 0 is. In fact, we're going to go even further back than that. And so this lecture, in addition to the podcast called Patient 0, will be for this module. Okay? And we'll talk a bit about the podcast later on, exactly where to start and where to finish. Now, so the basis of this particular lecture is to give you background information for the podcast. So the podcast is really extremely well done, and when I first heard this years ago, I had to make this lecture because it was such an interesting story about how this virus came to be and how it infected 80 million people that I wanted to share this with you. It is really an amazing biological story. Okay? Now, let's begin patient 0. Let's start off with this quote by James Locklove. An inefficient virus kills its host. A clever virus stays with it. And I told you this at the beginning. If you want to be one of the ultimate in terms of pathogens, you take out the immune system and you stay with your host for a very long period of time. And that's what the HIV virus does. So in terms of the infections, you'll see that this particular slide shows data from about 2015. Well, it really hasn't changed altogether that much. I checked out the 2018, 2019 numbers, and we're hovering around the 37.9 million people, so close to 40 million. In addition, we have about 18 million that are being treated for this disease worldwide. And unfortunately, we're still having people die from this disease on the order of about 1 million per year. And about 1.7 to 1.8 million people are being infected on an annual basis. So in sum, we have about 80 million that have been infected with the disease, and about 40 million have succumbed to this disease. And when you start to hear the podcast, there's a couple of definitions that you have to be aware of, and I'll go through those as we go through. One of these definitions is is the word spillover. As you know, the word spillover essentially means that this virus had an animal source. So HIV, the infection that causes HIV in humans, is really a zoonotic disease. And we use the word spillover, or sometimes you hear the word crossover, but I prefer the word spillover because it has more dramatic effect. Spillover from in this particular case, it's going to be a primate into humans. Now, in order to give you the background information for this particular part of the lecture, we have to introduce the major players. And here they are. What I tried to do with this image is to bring out the cutest possible images I could find on the internet. Why? Because I want to invoke basically a emotional response from you, because the development of this virus, specifically the SIV, the simian immunodeficient virus, has taken place over probably millions of years. And what I want to get into is how virus came into being. This is the SIV from chimpanzees. So, the players that we're going to be talking about: 1, the red-capped mangabe; 2, the spotted-nosed guenon; and 3, chimpanzees. And in particular, it's going to be a very special subset of chimpanzees, and we'll get to that in a little while. But what I want to draw your attention to is the following. When we're looking at SIV, like we have labeled here, we have the short code for red-capped mangabe, and SIV for spotted-nosed guenon, and the SIV for the chimpanzee. Each of these SIVs are different from each other. They're specific for that specific primate. In terms of the number of SIVs, there's at least 40 different types that infect different monkey and primate types. When we're looking at the one that infected humans, it came from this SIV chimpanzee. And somehow, this SIV made the spillover or crossover into humans and became HIV-1. We're not going to get into HIV-2 or any of the other ones, but what we're going to talk about is the main ones that infect humans. And we'll see how the SIV chimpanzee developed. So what is spillover? Well, essentially, you have to have some sort of exposure to the virus. And this occurred in a very special place in southeast Cameroon. And it occurred probably, and I'm going to say probably because we don't know the exact answers yet, probably occurred somewhere around the late 1800s. The spillover occurred, and then we have spreading of this said virus. In terms of the spillover in development specifically of the SIV chimpanzee, this virus was the merging, if you would, or the recombination of at least 2 primate species. Half of it comes from the red-capped mangabe, and the other half comes from the spotted-nosed guenon. We suspect there could be a 3rd virus, SIV virus, involved, but we don't know the details of that specific virus as of yet. So in terms of forming a hybrid, and we'll show you the genetic information in a little while, this virus, the simian chimpanzee virus, this SIV chimpanzee virus spilled over into humans, made the leap into humans, actually several times, and we'll go into more details about that. And the major one that we're going to focus on is the HIV-1M subtype. This is the mosaic structure of the SIV chimpanzee genome. Now, the important features are as follows. So we have the beginning and the end of the genetic information. It's about 9 kilobases in length. The first part in gray that you see here is from the SIV from the Rebcap-Mangabe. What's labeled in black, we're showing here, is from the spotted-nosed guenon SIV. So the chimpanzee genome that we see here is a hybrid molecule forming part from one monkey and part from another monkey to form the simian SIV chimpanzee genome. So this is the actual viral genome that we're showing you right here. Back to the story about the origin of the pandemic. We stated earlier that this disease first started in southeast Cameroon in a very, very specific type of chimpanzee. In reality, it's almost what we call an island effect. Why? Because these chimps were contained within a very small area of southeast Cameroon. Why? Because there's a series of rivers that essentially lock this geographic piece of land to about 100 square kilometers. So we have a river going up this way, a river going down this way, and another river going up into this area in here. So these chimpanzees are essentially locked within this given area. And essentially, this is where the actual virus was developed, stayed within the chimps for a very long period of time, and in this area here, this is where the development of the real patient zero took place. And this took place sometime around the late 1800s, early 1900s. In terms of transmission, the transmission would follow patterns of migration that occurred back in the late 1800s, 1900s. And that are following the rivers. So here we have the Sahan River leading into, and it's a tributary of the Congo River. And so imagine that person who first contracted the disease essentially followed rivers for communication pathways. So they followed the river, it led into the Congo River, which led right into some of the major civilized zones within Africa. Kinshasa is one of the major cities, and this is where in the early 1900s, 1920-ish, this is where the basically epidemic started. In addition, when you're listening to the podcast, make sure you have this map and a couple other slides I'll show you later on available to you so you understand more of what's going on within the actual podcast, because they're going to be talking about this. Back to the HIV virus. So in terms of this particular virus, HIV-1, which will be the one that we're going to be talking about, there has been at least 4 major groupings of this virus. In fact, there's actually a few more that they're trying to discover now. Group N is the main subtype that we're going to be focusing on. There's Group N, O, and P, and we'll get to what these are very, very shortly. Within Group M, we have a variety of subtypes. We call these clades, C-L-A-D-E-S, A through K, and then we have what we call circulating recombinant forms. Now, in terms of the clades, these subtypes of Group M, the circulating recombinant forms are actually mosaics of these subtypes or clades. And we have a link to a website for the circulating recombinant forms that we'll get to in a minute. Let's talk Group M. This is the major group, hence the M. It accounts for basically all of the HIV cases encountered within the world. This is the major one. We have within North America, we have what we call clade B, and this is the predominant form that's found within North America, Europe, Japan, and Australia. These other subtypes, what we call the circulating recombinant forms, if you click on this link, it'll take you to a website that shows that there are approximately 102, I believe they're up to right now, 102 different circulating recombinant forms. Those are just very minor deviations from the actual HIVs. There still are HIVs, but they do deviate slightly in their effects. One of them, CRF number 19, is a rather more potent version of the HIV virus, and that's kind of scary. That particular one came out of Cuba, and normally, if you have a normal HIV infection with clade B, the demise time, if you would, or the approximate time for infection is approximately 10 years, and we'll get to more of that later on. With this circulating recombinant form number 19 out of Cuba, that lifespan shortens to about 2 or 3 years, so it's a very, very active virus. Group O, also called outlier, infects about 2% of the population from Cameroon, and is really not seen outside West Central Africa at all. So it's a very, very minor effect. Group N, oddly enough, refers to being non-M or non-O groups. And it was discovered in 1998, again in Cameroon, and there's very, very few of these infections that have been reported. Group P is the latest group. This one originated from the SIV gorilla, which came from chimpanzee as well. And at this stage of the game, it's actually infected only one person who's residing in France. So very little infectivity from this set. And we'll explore why specifically the main HIV actually was and how it essentially spread so very far. On to patient 0. So this information is important for what the podcast will be talking about. In particular, what you will see is, and they will talk about, is this map. And this shows basically patient 0 right in this area, and then like spokes on a bicycle wheel, how this infection spread over time. And so the podcast is going to refer to this, and it shows you just the numbers of cases that have gone geographically. And this is the actual map that they used. It was published in the American Journal of Medicine back in 1984 to essentially highlight patient 0. And this patient 0 that they're talking about is that Canadian. On to a bit about the actual nature of the HIV virus particle. From the shape of it, we can see that this is an envelope virus. It contains 2 identical strands of genetic information. In particular, it is plus-sense single-stranded RNA. It has 2 copies. There are a variety of different reasons as to why it has 2 copies. In fact, retroviruses are known to have 2 copies of their genetic information. And because they do have 2 copies, they are considered a segmented virus, even though the 2 copies are identical. One of the major reasons why they have 2 copies is because of recombination. And we'll get into a little bit of that later on. In terms of the disease progression, we have in this table here, we have hours, followed by days, weeks, and years. So the initial infection will occur. Usually it's a dendritic cell. Why? Because they're almost always going to be associated with the epithelial cells. And so they have the receptors for the HIV virus. So the HIV virus will go in, cross the barrier, and once the HIV virus gets into the connective tissue layer, which we designate as lamina propria, it infects a variety of different cell types, specifically dendritic cells, macrophages, as well as CD4 cells, T cells, T helper cells. This virus, once it enters the cell, and we'll show that in a little while, integrates into the host genome and is able to be dormant for many, many years. In addition, once a T cell has been infected, or other cells, macrophages, become infected, and monocytes for that matter, the virus has the ability to essentially make more copies and copies and copies of itself. So as your cells divide, this retrovirus also is able to divide, because it's basically it's integrated its genomic information into yours, and whenever you replicate, it replicates too. And as time progresses, we get more and more of the virus being disseminated, and we're talking days, so it's a very short cycle for this particular point. As time progresses even further, it's going to move to distant sites within your body. And one of the obvious choices is the spleen and lymph node. So here we have the infectivity going into the lymph node, where we see more and more immune activation. And as time progresses even more, we have this sustained HIV production. So this is years, and it can be up to 10 years in terms of response. And I'll show you that in a little while. So how does this virus really function? Please click onto the video and watch the video. The video actually doesn't have any sound to it, but it does give a good approximation as to how the virus actually replicates. The virus is highly complex in terms of how it functions. There's no way we can go through all what it can do. It's very, very complex. First thing it does, it's an envelope virus. Here is the genetic information surrounded by its capsid. So we have a nuclear capsid in this area here, surrounded by the envelope, which has the proteins that are necessary for binding to the actual receptors. So these are the keys needed to get into the keyholes in order for the virus to enter the cell. The virus actually needs two keyholes. We have a CD4 from the T helper cells in this area here. These are the CD4 markers. In addition, it needs a co-receptor, and the co-receptor is CCR5 or CCR4 in terms of the chemokine receptors. Once inside, this endosome splits open, and the genetic information, poof, spreads out. So at this stage of the game, the virus is said to be eclipsed. One of the first proteins that it comes across is the reverse transcriptase. Now, before that happens, you may have a question. The question is, why doesn't, since this is a single-stranded plus strand orientation RNA, why doesn't it go to act as messenger RNA? Well, it's capped at the ends, so it doesn't get into the ribosome cell. But reverse transcriptase can work on it, makes a double-stranded DNA copy, and this goes into our nucleus through the nuclear pore, and this is quite complex in terms of how it gets in. We're just telling it gets in, but in reality, it's really, really complex, using a whole series of proteins. Very interesting area of research. In addition, in terms of the life cycle, we have integration into our genome. At this stage of the game, we call it a provirus. From here, we can have a very long latency period. And again, as your cells replicate, this will also replicate as well, because it's now part of your genome. So we're getting more copies of it that way. In addition, as time progresses, we will have viral messenger RNA being produced, and we have the production of what we call late-stage proteins. First of all, you're going to get early-stage proteins. They tend to be more of the reverse transcriptase, etc., etc. One thing I forgot to mention, in order for this virus to be viable, it has to bring with it protein elements that are necessary for its replication. Specifically, it has to have brought with it reverse transcriptase, an integrase, and a variety of other proteins. In addition, it makes use of a piece of your transfer RNA. Sometimes that transfer RNA will be within the actual virion itself. Other times it can utilize transfer RNAs that are present within this area. As viral messenger RNA is being produced, we'll have early phase proteins, which tend to be more the polymerases and integrases, and then late stage proteins that will be produced. These usually are the proteins that are necessary for envelope proteins as well as the matrix proteins. And so as the virus gets close to being released, it's going to create what we call a lipid raft. And that consists of, within humans, we have a lot of cholesterol found within this area. And then this is, in the case of the virus, all the matrix proteins as well as the capsid proteins, spike proteins, will go to the cytoplasmic membrane. And the virus, the double-stranded genome, will make its way up to this raft, lipid raft, and will have budding out of the specific virus. And here we have a budded out virus, HIV virus, that will go on to infect other hosts. This is actually one of two major ways that this virus can actually infect cells. One of them is via the release of the actual virion to go infect other hosts. Another method is lateral, so going from one cell to another laterally. And here we have a very organized orchestra of proteins to move the virus to the sides of these cells, and they form what we call a viral synapse. And the virus from there pushes essentially its way into the adjacent host by merging of these cell membranes. And once it's in the other host, it acts and behaves as normal nucleic acid, and reverse transcriptase picks up on it, and essentially we have double-stranded DNA being formed, integrates the virus into the next host's genome. In addition, we talked earlier about how we have early phase proteins and late phase proteins. Well, some of these early phase proteins actually attack your innate response. As this all is happening in terms of the virus coming into the cell, as soon as it gets in, these are perfect areas to attack the actual virus. So your body actually has methods to deal with reverse transcriptase, to deal with integrase, to deal with production of the messenger RNA. So the entire life cycle is an amazing way to combat this particular virus, and all viruses for that matter. When you look at the virus life cycle, you can hit anywhere within the virus life cycle in order to develop strategies to stop the virus. In this particular case, hitting the reverse transcriptase is pretty good. We have drugs that are available to inhibit reverse transcriptase, and they're so well done in terms of the drugs plus other drugs that we can take, that we can actually have a case where this entire disease is, I don't want to say cured, but ultimately patients have zero titer of viruses. That basically means they have zero levels of this virus within them. So nowadays you can actually live a very long life, a full life, if you are infected by taking these potent drugs. Now back to the early stage proteins. We actually have two major viral proteins that we're going to talk about later on that actually are produced, in one in the as an early stage protein and one as a late stage protein. NEF, which stands for negative factor, is an early stage protein, and that protein helps to combat your innate response, and more on the innate response that we do later on. VPU is a late viral protein, and that stands for viral protein ubiquinase. Oh, one correction, not ubiquinase, it's unique. Viral protein unique. Anyways, this late viral protein VPU also targets your innate system, and specifically it goes after specific proteins. One of the proteins that you produce in order to combat this disease is called tetherin, and we'll talk more about tetherin later on. Tetherin, as the word suggests, tethers the virus. It blocks the release of the actual virus. Both NEF and VPU target tetherin, especially in primates, some of the primates. In humans, VPU turns out to be a very potent anti-tetherin molecule. In humans, NEF lost that capability, but it retains other functions. In fact, whenever you're looking at these viral proteins, specifically the ones that go after your innate system, they actually have multiple targets. And that's one of the major strategies of this virus. When it produces these proteins have multiple targets that it uses to affect your cells. And we'll talk more about that as we progress through. To sum up, when we're looking at the HIV virus, you have to have that reverse transcriptase. And it goes from makes double-stranded DNA from single-stranded RNA. In other words, reverse transcriptase is an RNA-dependent DNA polymerase. Once we have double-stranded DNA, integrase is able to take and insert that double-stranded DNA into our human chromosome. We call that a provirus. And this basically allows through replication the propagation of this inserted genetic information. This provirus can be remain dormant, for many, many years. You'll see that in a graph later on. Macrophages, monocytes, dendritic cells, and essentially T4 helper cells can be a reservoir of HIV. We talked about the lipid rafts that it can take in order to escape its cells, but don't forget about that viral synapse. And that is a very interesting mechanism by which HIV can transfer from one cell to the adjacent cell beside it through that viral synapse on its lateral domains. Very interesting. And in the pathogenicity, HIV essentially destroys the immune system. Specifically, it takes out T4 helper cells and as well as macrophages and dendritic cells. And we'll talk more about that as we progress through. In terms of the course of action that the HIV virus follows, we have for example, up here we have our primary infection of the virus. So here's the number of HIV we have in the blood. As time progresses, it increases and then decreases. So what we see here in terms of the primary infection, we're looking at the levels of CD4 T helper cells. They drastically go down in the beginning, and then they have a slow rebound as they go up. So we have decreasing HIV numbers, increasing CD4 T helper cells, as well as antibodies against that virus. Everything looks good. We're increasing. We seem to have a control of the disease. The number of virions is decreasing. But then over time, we have a reversal of the situation. So the immune system is now compromised, and we see decreasing numbers, increasing numbers of the HIV virus until death. Because T4 helper cells, as well as the amount of antibodies against it. And don't forget, antibodies are being produced by B cells. Okay, B cells that differentiate into plasma cells. So these B cells and plasma cells start to decrease as well. Their numbers go down. Until ultimately, about a decade later, we have the death of the patient. Now that circular recombinant form that we talked about early, CRF19, that came out of Cuba, imagine that curve squished into about 2 or 3 years. So dramatic differences in the activity of some of these viruses. So what does this all mean? Let's talk about how we combat the HIV virus. There's a couple of mechanisms by which this happens. When you're looking at the genetic code and you're looking at the protein coats that this virus produces, one of them is what we call a GAG protein. That stands for group antigen proteins. And this essentially makes up the receptors that sit on the surface of the actual virus itself, on the envelope. One major change that occurred within GAP protein number 17 occurred at position number 30. And we have a change from MET to arginine. And that change, that simple change, in addition to having a high degree of VPU activity as an anti-tetherin molecule essentially allowed this virus to propagate within humans. Within chimpanzees, that gap at position 30 of protein 17 has to be methionine. When we inject chimpanzees with human HIV, there is little effect. In fact, the genetic pressure on that gap protein number 17 at gap 30 position, is so intense that it actually reverts back from arginine to methionine. So there's really a story to be told there at some point. Very, very interesting. In addition, we have the two major anti-tetherin proteins. And this is how they function. There's actually a third one of the environmental proteins, but it really doesn't play much within humans. We're just going to discuss NEF and VPU. And remember, NEF is an early phase protein, and VPU is a late stage protein. How do they function? Well, in terms of NEF, NEF works very effectively in certain primate species, specifically the red-capped mangabey. NEF binds to and interacts with and binds to the cytoplasmic domain of our tetherin protein. So NEF is a viral protein. Tetherin is the protein that we make that essentially tethers or latches onto the virion as it escapes our cells. And essentially, once tetherin binds to the HIV virus, it locks it in. So it cannot bud out. More on that in a little while. NEF binds to the cytoplasmic domain of tetherin, preventing it from attaching to essentially the virion particle. That works very well and very effectively in some primates. In humans, we have a 5-codon change within our cytoplasmic domain of tetherin. So NEF within humans, NEF does not bind to tetherin. So within humans, NEF has very little to zero anti-tetherin activity. But NEF does have other capabilities, and we'll talk about those in a little while. It is a very potent way of dealing with other molecules on the cell surface. We'll talk more about that in a little while. VPU binds to the transmembrane domain within tetherin. And essentially, once it binds, it can take tetherin out of action. So where did all this come from? As we can see here, this is the HIV-1 group that caused basically the pandemic that we're seeing. It was able to grab NEF from a specific monkey and VPU from another specific monkey. So the spotted nose guenon gave us VPU, and NEF came from the red-capped mangabey. This was into the SIV chimpanzees, and this essentially became the HIV-M group that caused the pandemic. In terms of humans, this NEF is ineffective because we have the 5-codon deletion within the cytoplasmic domain of tetherin, which essentially made NEF not function as an anti-tetherin molecule. In our case, it's VPU that became a very potent anti-tetherin molecule. More on that in a minute. In terms of this figure, we're going to show you how VPU, and to a limited extent, how NEF operates. So in absence of VPU, in other words, we have tetherin working the way it's supposed to. We have tetherin. In this particular case, tetherin is BST2. Tetherin also goes by a couple of different names. BST2 is one, and CD317 is the other. You don't have to know those completely. My advice is to understand tetherin as tetherin. I think that's the easiest way of working with tetherin. So normally, tetherin goes through the trans-Golgi network, goes up to the cell surface, binds to the molecules, the virion molecules that are being released, causes them to endocytose, go back into our cells where they are slated for degradation. Sorry about the vibration. Anyways, this is how normally our innate system responds to the virion, the HIV virion. So by definition, tetherin is an innate molecule that we produce to combat the HIV virus. It tethers, attaches to, preventing the budding out of these viruses, brings them back into the cell for degradation. In presence of VPU, which is a late-stage protein, tetherin does a couple of things. First of all, it interferes with membrane trafficking. That's key to the functionality of tetherin. Functionality of VPU. In this particular case, we're in the trans-Golgi network with VPU binding to tetherin within the trans-Golgi network. As it's being released as an endosome, it actually doesn't necessarily reach the cell membrane. From here into the endosome, it's scheduled for degradation by your normal lysosomal degradation pathways. So in this particular case, this tetherin molecule never reached the cell surface. If there are tetherin molecules on the cell surface, they can be bound to VPU and brought into the cell via endocytosis and again led down the lysosomal degradation pathway. So you're essentially eliminating tetherin from this particular cell. That's huge. So VPU is interfering with normal membrane trafficking of proteins. NEF also does something similar to this. It interferes with normal protein trafficking, except for tetherin. NEF doesn't have any anti-tethering capabilities. But like I said earlier, these molecules, these proteins that are being produced, have multiple sites that they can hit. One of the reviews I put online talks about VPU and tetherin, and specifically the membrane trafficking. I highly recommend that you read this. The title of this particular review is Remodeling of Host Cell Plasma Membrane by HIV, NEF, and VPU. It's a very, very detailed article. And when you read it, you will find out that NEF and VPU have numerous molecules that they bind to. Let me read you some of the more important ones, and we'll essentially see what happens. So NEF, CD4. It eliminates CD4 from the surface. So essentially what you're doing, following a similar pathway that's outlined here, NEF binds to the CD4 molecule, brings it in for degradation. So you're essentially erasing the identity of CD4 T helper cells by getting rid of the CD4 molecules. MHC1, MHC2, these are key molecules important for the immune system. These are immunoreceptors, important for antigen presentation. They're found on macrophages and other cell types. By taking those into the cell for degradation and stopping them from elimination even before they get to the cell surface, you're eliminating the identity of those specific cell types. So the question is, does HIV kill the cells? Yes, it does. It can. It does have that ability to do that. But in addition, it doesn't necessarily have to kill the cell. You can actually erase the identity of those cells by essentially interfering with membrane trafficking, eliminating those proteins, even before they get to the cell surface. So NEF interacts with about 10 different proteins. VPU does something very, very similar. VPU takes out CD4. Again, that's an incredibly important molecule that's necessary for your T helper cells to function. Tetherin is the other example that it takes out. And if you look at the table, there's approximately 10 more different types of proteins that it gets rid of. And we won't go through the list, it's too extensive. But the idea here is that we have two viral proteins that are geared towards membrane trafficking that are critical for the survival of this protein, of this virus. And not only is it critical for the virus survival, but it interferes with our cells' ability to combat this virus. And that's one of the major take-home messages. So obviously, this would be a major area for us to target. If we can target VPU, if we can target NEF, plus reverse transcriptase enzymes and other areas that are important in terms of viral survival, we can actually deal with this virus very, very effectively. The problem is getting us to design those proteins in order to combat them. That's the challenge. To sum up, we have our monkeys that are coming together to form the chimpanzee. This is basically the SIV, Spontanus guenin, red-capped mangabey, and another monkey we're not 100% sure of, to form up this adapted SIV chimpanzee, which means that it's now going to be very infectious towards not only the monkey, but in terms of the spillover effect into humans, we have, in terms of this specific genome, which is the one that infects humans, we've lost that NEF anti-tetherin activity, but it has retained the ability to deal with other molecules, and that's so critically important to understand that NEF may have lacked anti-tetherin, but it can target other proteins in terms of membrane trafficking. We have this gain of VPU-mediated anti-tetherin activity. In addition, it also, via membrane trafficking, interferes with other proteins too. And we have this third ideal, which is the GAP30 protein. So at position number 30 within protein 17 of the group antigen protein, it's somehow made it more efficient for the virus. In reality, it's the combination of these three plus a few other factors that makes this virus so lethal. So we need this mutation. It seems to enhance the activity within humans. VPU, incredibly important in terms of acting as an anti-tetherin. So in terms of us combating this disease, the two major ways, VPU. Anti-tetherin, the ability of tetherin to take out this virus is paramount. So whatever is going to interfere with that, whatever strategy the virus has to interfere with that, will make it more active. So this mutation, VPU, and limited extent is the NEF, but it's still important because NEF, although it's no longer an anti-tetherin molecule, does in fact help with the infectivity process in the early stages. It's going to start to erase out all the CD4 markers, MH1s and MHC2 molecules in order for these cells to no longer be functional. That concludes the lecture for today. Now on to the podcast. Now the Patient Zero podcast actually consists of 3 parts. The first part goes into cholera. Specifically, we're talking about Typhoid Mary. You can listen to that if you wish. It's actually very interesting. But if you wish to skip that, Patient Zero, in terms of the HAV, begins at the 16-minute and 5-second mark and goes to about the 44-minute mark. So it's about 35 minutes in total length of time. The third part of that podcast is actually about Ebola virus, and that too is quite interesting. So if you wish, you can listen to that too. But if you just want to go with the Patient Zero in terms of the HIV virus, you can start at 16 minutes and 5 seconds. Start there and then continue from there. Okay? Thank you very much, and I will post this lecture as soon as I possibly can.